Recombinant DNA technology. One of the key technologies used in the research and manufacture of biopharmaceuticals is recombinant DNA technology. This technology allows us to produce proteins outside of the body, that is, in an industrial setting, in large quantities. All organisms use the same genetic coding system to make proteins. In other words, they use genes. It is possible to identify and isolate specific genes that instruct cells how to make specific proteins. These genes can be taken from one type of organism and introduced into the genetic material of another organism. The second organism can then be induced to produce the required protein. In the case of recombinant insulin, the human DNA for making insulin is inserted into the DNA of E. coli, a simple type of bacteria. The bacterial DNA then becomes modified so that the bacterial cell will produce the human insulin protein. Because this process involves recombining the DNA of two different organisms, it is called recombinant. The manufactured insulin then performs the same function as naturally occurring insulin in the body. The organism that has its DNA modified is known as the host, and the actual protein produced is called the expressed protein. In mammalian cells, many proteins undergo extensive modification following translation. Many of these post-translational modifications can only be performed by mammalian cells. Bacterial cells lack the required cell infrastructure needed to perform complex post-translational modifications of mammalian proteins. This means that mammalian cells must be used for producing more complex therapeutic proteins.